So, hey everybody, my name is John. Really not necessary. I'm a graduate student. I've been funded by BLAST for the last year and a half now. And what I'm going to be talking to you guys about today is data analysis. It's sort of going to be a bridge between, okay, you've done your experiment, you have some data, and maybe what are the next steps? What do you do with it? What I'm not going to be going into too much is hardcore statistics, so I'm not going to be teaching you Stats 401 right now. Hopefully you guys have taken that or will take that at some point during your degrees. Or there's a really good person who's sitting in the back right now as a resource for you guys. That's Andres, for you guys that don't know him. He is an amazing statistician, and he is available for you guys to consult if you have any data questions. And he knows a lot more about stats than I do, so if you have any statistical questions about specific tests or which tests to run, he would be a great resource for you guys there. So, start off the presentation with a quote from Adam Savage, who, as you guys probably know, is one of the Mythbusters. And he says, "The only difference between screwing around in science is writing it down." And I would say this is very true, especially if you happen to like your project and like what the science that you're doing. A lot of times it can feel like a lot of fun. You get to, put things in a test tube and see if it explodes, or you get to, play with some really dangerous chemicals. And if you don't write things down, you're not doing science. It's really, really hard to do good science without keeping really, really detailed notes. And so I'd highly encourage you guys, when you're doing your experiments, just to write everything down. Even if you don't think it's important now, it might be important later, so just write it down. I'm going to start off with a brief video that I shamelessly pilfered from YouTube. So this video is kind of addressing what is data. And let's see if the sound works here. Works really well. It's got some cool funky music.
This short library video will introduce you to the basics of data. You'll learn what data is, why we use it, and how it is collected. Data is everywhere. It is a collection of facts. There's qualitative data, the textual description. The latte has a robust aroma, frothy appearance, and strong taste. It's in a white cup. There's also quantitative data, referring to a number. The latte contains 12 ounces of drink. It's served at 150 degrees Fahrenheit and costs $4.95. There are two different types of quantitative data. There's categorical data that puts the item into a category. For example, if we have a box of shirts, we might collect shirt size data with categories such as small, medium, and large. There's also continuous data. This is numerical data that can take on any value in a certain range. For example, height. So why do we use data? We use data for different reasons. Data can be used to answer a question or tell a story. Having a question to answer can provide focus and make it easier to find patterns with the data. For example, we might use data to answer a question of, do students who spend a lot of time on social media have poorer grades? When telling a story, we may not always have a question in mind, but there might be a story hidden within the data. When telling the story of climate change with data, we might use global temperature patterns, weather cycles, and greenhouse gas data. How do we collect data? We collect data from primary and secondary sources. Primary sources might include data collected by researchers through interviews, observations, case studies, questionnaires. Secondary sources might include data that already exists: official statistics, web information, government reports, previous research. Need help? Ask us. Chat, email, phone, or drop by. video on the basics of data.
So I thought that was a nice little video kind of giving you guys an overview of data. A lot of people might not realize when you're talking about data, some people just assume that it's always going to be a number. It's always going to be, you know, a concentration or some sort of continuous variable. But data can be anything. Data can be, you know, a description. It can be a qualitative variable. It can be some sort of categorical variable. Also another reason why it's really, really important when you guys are collecting your data to write things down. So sometimes when you're out in the field doing an experiment and you collect data on a Monday and then, you know, Tuesday, Wednesday, Thursday, and then you go back and you look and you see this weird pattern where all the data you collected on Monday, you know, is really different than all the other data. It might just be that, you know, maybe you're really sleepy on Monday, and maybe you're not just with it and you're not collecting good data. And so writing something like that down really helps you analyze your data later. So I wrote up for you guys in a handout basically a flow chart, if you will, of what to do once you collect some data. And you'll notice that in the flow chart, actually conducting the experiment appears rather late in sort of the flow of ideas for experimental design, collecting data, analyzing data. You know, doing the experiment, analyzing the data comes really late in all of that. So the steps leading up to that in an ideal world would be to first specify a biological question. So you go out into nature, you know, in nature in this case could be a plate, you know, what you're doing a microbial culture on. But you make some sort of observation. You say, you know what, I think that coniferous trees in this region are going to be taller than deciduous trees. That's a great observation. Put that question in the form of a biological null hypothesis. So how many people have taken Stats 401, some kind of statistics class? And have you guys taken other stats classes here at UAF? Stats 200. 200? Okay. Yeah. So in those classes, they talked about things like probably like, you know, comparing means, you know, two sample t-tests, linear regressions, things like that. Those are all really, really good statistical tools, and they're things that are very, very common in science. So you guys probably heard the term null hypothesis before, right? Can anybody explain what a null hypothesis is?
Basically, if the data has no meaning, like the data, there's no linkage between this variable and another variable.
Oftentimes, yes, that's what it is. So if I have two variables and I say that I think that, you know, you know, deciduous trees are going to be taller than coniferous trees in this region. A null hypothesis for that assumption or that observation might be that there's no difference between these trees. And the reason that we do that is actually because when we do statistics, a lot of times what we're trying to do is we're actually trying to disprove our null hypotheses. And so a lot of times we specify a null hypothesis as being that there's no difference or that there's no change. But that's something that Andres can help you guys with a little bit more. So after you form a biological null hypothesis, you come up with a statistical null hypothesis, saying like there's going to be no difference between these tree types or that there's no relationship between these two variables. Determine which variables are relevant to the question. If I'm trying to determine what's higher, a coniferous tree or a deciduous tree, something like soil type is probably not going to be that important to my question unless I think that soil type is the thing that contributes to the height of trees. Something like, you know, daily mean temperature is probably not going to be a variable that I'm interested in. I can collect it, but will I end up using it? Is it relevant to my biological question of which tree is higher? Probably not. Determine what kind of variable each one is. So whether your data are going to be a continuous variable, whether it's going to be a categorical variable. Something like tree height would be a continuous variable, right? Because it would be, you know, height off the ground. You could express that in meters or centimeters or whichever unit of measurement you'd like. But it could also be a categorical variable. If you're not particularly good at, you know, trigonometry or measuring trees, you could say, I think that tree is tall, or I think that tree is short, or I think that tree is medium height. And as long as you had some sort of bins that you could put those trees into, that's a perfectly valid way of analyzing that question. So design an experiment that controls or randomizes confounding variables. You don't always have to do an experiment in science. A lot of science is observational. So going out and measuring how tall trees are, you're not necessarily conducting an experiment. That would be considered observational science. You're making observations about the natural world. But a lot of you guys will probably be doing experiments where you're saying, I'm going to expose this mouse to this chemical and see what happens, or I'm going to grow these two bacteria on these different substrates and see which one does better. You want to design an experiment that randomizes these variables, and you want to do one that minimizes any potential confounding variables. Does anybody know what a confounding variable is? Not necessarily an outlier, although it could lead to outliers. Anybody? Brandon? Got anything? So confounding variables are variables that you are not particularly interested in. They're not addressing your main scientific question, but they could throw off your data. So if I'm interested in seeing what's taller, deciduous trees or coniferous trees, and I go to 5 different places all around Fairbanks, and one of those places happens to be, a swamp or one of the lowlands. In those places, all there is black spruce trees, and so I would find that my black spruce trees are higher than anything else just because there is no deciduous trees in that region, right? So the soil type in that case would be a confounding variable because the soil type is leading to the height of the trees. It's not necessarily something that I'm interested in. I don't really care why the deciduous trees are taller than coniferous trees. But in that case, soil type is actually leading to the black spruce being taller than anything else. So confounding variables are things that we're not particularly interested in. They don't address our biological question, but they are something that you could collect data on. But you want to try to control them when you're designing experiments. So based on the number of variables, the kinds, a lot of yada yada, choose the best statistical test to use. So this is where, again, Andres can help you. But you notice that in an ideal world, you've actually chosen your statistical test before you do your experiment. How often do you think this happens in real life? Not very often. Most of the time, you end up collecting your data, you do your experiment, and then you think, okay, what kind of statistical test can I use? But in an ideal world, you've thought about this already. You've thought about your experiment, you've designed an experiment on paper, and you've said, okay, once I collect these data, I'm going to be able to use this statistical test to make this comparison. And that's where, when you're doing all of this experimental design, you're designing an experiment, it's really, really good idea to sit down with a statistician before you even do the experiment, and they can tell you whether or not your statistical test that you've chosen is going to work. Whether or not it's going to be able to eventually tell you if there are differences between these two tree types. And whether or not your experimental design will actually meet the assumptions of that statistical test. So if possible, do a power analysis to determine what's a good sample size. I'll be talking a little bit about power analyses in a minute, but they're generally a good idea. And actually, if you guys are doing, I don't know if anybody here is doing work with lab animals. Yeah? If you're doing work with vertebrate lab animals, those are covered by IACUC, or the International Use and Care of Animals Committee, and those actually often require you to do a power analysis before you do an experiment. As they want to make sure you're using the minimum number of animals possible so that we minimize our impact on lab animals. So all the way down here now at number 9, we actually have do the experiment. So that's 8 things that we are supposed to do before we do the experiment. So after you do the experiment, examine your data to make sure that it meets the assumptions of your test. So a lot of times the assumptions, especially in parametric statistics, we have normal distributions, homoscedasticity, and if our data don't meet those tests, then we have to go through and go back and find an appropriate test, or we have to figure out what went wrong in our experiment, why we didn't get those tight distributions. So number 11, apply statistical tests, and 12 is communicate the results. So make a poster, give a talk, write a paper. So that's the ideal world. And you'll notice here we have do the experiment. Oh, well, that's supposed to be around do the experiment. We have choose the statistical test before we do the experiment. That's an ideal world situation. In the real world, it's slightly different. So generally what happens, and at least as a grad student, is that your advisor drops you off in the middle of nowhere for an entire summer. So he says, here's a field camp, go collect some data. You have no idea what you're doing out there. He tells you to measure everything. So if you're out there and you're supposed to be measuring tree height, he says, measure tree height, give me soil salinity, give me bacterial cultures, give me average daily temperatures, wind speed. You're just measuring everything. You get back to the lab and you realize that all of your samples are contaminated or you have way too many confounding variables. So you sit down and you say, okay, all of this was fine, but I'm actually going to sit down now and design an experiment. So generally you already have done the experiment when you sit down and you say, I want to design the experiment correctly this time. So then you head back out into the field for the next summer and you collect your data again. And this time you have a plan. You got the variables that you're going to measure. You know how many times you're going to measure them. You know what degree of certainty you want to get for each variable. So you look and you say, okay, I know what my confounding variables are. I know that this variable is weird. I'm just going to ignore it. I don't care what it's telling me. You make some cool plots. Those are nice. You try to figure out which statistical test you can use after you make the plots. And you decide on a t-test because it's easy, and that's the one you're most familiar with, so you try to make a t-test work. You check and see if your data meet the assumptions of a t-test. They don't. They usually don't. So you go back to step number 9, and you try to figure out another statistical test to use. And you repeat this as often as necessary. Then you go ask the statistician for help. And you come to them and you say, no, my data don't work. They don't meet any of the assumptions of the tests I'm trying to use. And they cry, and then you cry softly as they pick apart your poorly designed experiment. So, what I want to highlight here is that it's a really good idea to talk to somebody who's familiar with statistics. If you're not personally familiar with statistics, go talk to them before you design an experiment. If I had known that my first year, I probably would have saved an entire year of my PhD research, just because I'd ended up designing an experiment where I couldn't rule out some confounding variables, and it ended up being a huge waste of time. So go talk to somebody who's familiar with stats before you design an experiment, and make sure that what you're doing is appropriate and right, and make sure you guys come up with a plan for your statistical test to use. So you'll notice in this plot, that should be slightly up. Statistical test comes after you collect your data. That's the wrong way. Wrong way to do it. Decide on stats, then collect your data. So I would highly encourage you guys, if it's not required by your program already, to take a course in statistics even before you do an experiment. I know some of you guys have taken stats, maybe 200, 401. That's great. That provides you with a little bit of foundation. You can start thinking in that statistical mindset. I know probably not all of you guys are experts in statistics. I know I'm not. And so at that point, you can say, okay, I have a rough idea for how I want to do this experiment, and then you can take it to somebody like a professor or a graduate student or Andres, who is more familiar with statistics, and they can help you refine the experimental design. So talk to a statistician. I have a little section on here on how to avoid some common mistakes in sort of data collection. So the first common mistake is you don't write everything down. So this was actually an experiment that I did where I was looking at 2 treatments, so treatment 1 and treatment 2. And then this is my variable that I was interested in. It doesn't really matter what it is, but you can see that it looks like there's a significant difference here. One of them is much higher than the other one. And I was like, great, treatment 2 is awesome. It's significantly different. But what I neglected to think about was that one of these treatments I measured only on one day, and then the next day I came in and I measured them. I measured all of the treatment 2 samples on a different day. What had happened was the machine overnight had totally recalibrated itself and was now spitting out completely different numbers. And so the things that I measured on day 1, things like our quality assurance, quality control samples, were totally different on day 2. And so based on that, I could not trust any of those data because our quality assurance got thrown out the window. I had to totally ignore that my stats were telling me that there was a difference between these 2 things. If I hadn't written down what day I actually made these measurements on, I would never have seen that. I would have just said, hey, there's a statistical difference. I can make a pretty plot and show you guys that there's a difference. But none of that was real. None of that was valid. And there's a number of instances where writing things down, like what day you measured something on, what the machine was running at, what your gas was flowing at, writing things like that down have saved me a couple times from coming up with some bad data. So definitely write things down in your lab notebooks. The other thing a common mistake is to not play around with your data a little bit before trusting your statistical tests. Not examining your data. I call it through kind of wading into your data a little bit. it's oftentimes maybe a good idea to make a couple plots to kind of explore your data before you go through and do your rigorous statistical tests. Because if you had done a test, for example, a linear regression test on these data here, you would determine that there's probably a significant relationship in these data. It looks like there's a strong positive correlation between these, or strong positive relationship. But as you guys can see from when we actually plot the data, if we got rid of that little data point, the relationship would basically be nothing. It would be a complete spread shot of data points, and there probably would not be that strong positive relationship. And what you need to think about is whether or not that data point is an outlier because you screwed something up, like you measured it wrong, or somebody jostled your arm while you were pipetting, or you just forgot to add something to a reaction. Whether it's due to a human error, in other words, if it's an actual biological phenomenon, like if that data point is real and you didn't screw up the data, you have to think long and hard about whether or not you want to try to remove that before you do these analyses. But if you try to publish something like this in a scientific journal and say, hey, there's a really strong positive relationship, it will probably not be published. The other thing that you can see sometimes is you can see if we have a really strong tight relationship between two variables like this, and then we have a few outlying points over here, we can see that those are throwing off our regression curve by quite a bit. These are what we would call influential points. They're not really outliers like this one is. So this is an outlier. You guys probably heard the term outlier. Outlier means it's way out. Like it's off. It's away from our means. It's away from our couple standard deviations. These, if we plotted them, they're well within the min and max of these other variables. So the max of this y-axis would be here, the min of the y-axis, and then the min of the x and the max of the x. They're well within the range of our other data points, but they're influential because look at how much they change the regression. So this red line is if we include those influential points, it totally changes the regression line. So it goes from being a really good tight fit to being a kind of sloppy and not so tight of a fit. And again, it'll be up to you guys to determine whether those are real observations, whether your data is just kind of noisy, whether there was something that went wrong with those particular data points. You can exclude them based on something went wrong in a reaction or something went wrong in your analyses. So examine your data before just blindly trusting your statistics is the take-home message here. Play around with it a little, make some plots. Another common mistake that I see a lot, especially with with PIs, I'm glad there aren't any in here right now, but they seem to really like, one, doing statistics and plots in Excel, which is not an ideal situation. It is doable, it's totally valid, but it's not ideal, I don't think. But also the data that they keep is just kind of messy. So you'll notice here that we have a, it's kind of hard to read, but we have a column called mutant, and then there's a bunch of just blank spaces in here. There's like blank columns, there's some columns, there's like weird columns with kind of weird calculations, but not all of them are being calculated properly. There's a plot just in the middle of the spreadsheet. This is not what I would call a clean data sheet. And when somebody goes back and they look at they look at this data sheet, if somebody was trying to replicate your study, they would be like, what even are these columns? There's no column names. They're not labeled in any way. It's just a string of numbers. And if you went back in this, the day after you did your experiment, you'd be like, oh, this makes total sense, But I guarantee you, if you went back to this in 3 years and you tried to figure out what was going on, it would be much, much harder. So always, have like column headers, maybe even have like another spreadsheet or another like Word file that's associated with this spreadsheet to give a little description about what each column is, where those data come from. And having your data in a more clean format will greatly, greatly assist you if you're trying to run statistics in programs like R or Python or SPSS or something like that. So keep your data clean. That's bad. Don't do that. Something like this is good. So here we have it's a very simple spreadsheet. There's no like colors. having things like, color designations and different color columns is not necessarily bad. But it's very easy to keep a simple, clean spreadsheet like this. We have an ID for an animal, which island it came from, what year it was collected, what sex, mass, length. Everything's all filled out. There's no weird spaces in between your rows, spaces in between your columns. Each of the column headers is descriptive. I can tell what it is based on what the column header is. So this would be an example of a good workbook or a good worksheet, good way to keep data. Now this is not what your lab notebooks are going to look like. Lab notebooks are a whole different animal. Lab notebooks generally are pretty messy because you're trying to write things down very hastily, and you're not going to be able to keep things in a nice spreadsheet like this. But when you do end up entering your lab notebook data into your computer, try to keep it clean, keep it looking like this. So there's a lot of different software when it comes to statistics. When you guys end up doing your data analysis, there's a few different options for things to choose from. The one I don't have up here, which is a possibility, is working in Excel. So you have all of your data kept in Excel. Excel does do statistics. There are statistical functions. You can do things like t-test and ANOVAs and linear regressions, and that is definitely an option if that's something that you're familiar with and comfortable with. But by and large, most people who are publishing scientific papers are using one of these programs. So I put this little table up here. I'm not going to go through it cell by cell, but there's a couple things I want to point out. So all of these programs have some sort of pay to play or licensing feature associated with them. If you're at the university here, you can get a lot of these for free. I know you can get SAS and Stata for free. Just through, if you download it through the university. We have a license for it, so they just, you can download it through the university and you can use it anywhere on campus. I don't think we have a MATLAB license. MATLAB is a very specific, I found that it has sort of a limited scope. It does some things really, really well. It does signal processing really, really well. It does satellite information, satellite tracking really, really well. But it's fairly limited in what it does. So unless your lab uses a machine that can only be analyzed through MATLAB, I would try to ignore that one. It's something that if it works really, really well for a couple things, but if your lab's not using it, or if you don't want to take the time to learn it, I would give that one a pass. So if you look at the total number of citations per year, the science in general is actually kind of it's dominated by SPSS and SAS. So that's the red and the green lines up there. Those two have been sort of the most used over the last decade. So this scale is from 1995 to 2015. So the last 2 decades. Those two have definitely been the most that have been used over that time frame. They're pretty intuitive. They're simple to use. A lot of them are point and click. So you say, okay, I'm going to import my data, and then you click and you say, I want to run a t-test between this group and this group, and then it spits out a p-value for you. So it's pretty intuitive, pretty easy to learn. However, you'll notice that both of those programs are showing a pretty sharp decline over the last 5 years. And if you look at the change in articles published from 2014 to 2015, so over the course of 2 years, you'll notice that both SAS and SPSS have dropped by about 25 or 30%. And there's a number of reasons why this is. One of the main reasons, I think, is that these programs up here, by and large, are free. They don't cost any money. They're free to use, and they're open source. And because of that, there's a huge community that develops around those programs that is there to basically help you and help each other develop packages and develop software based in those programs. So a program that a lot of people use these days is R, the R programming language. It has an enormous functionality. It has packages that are developed by software engineers and by biologists and people who have developed these packages in a way where they have functions that are extremely useful for analyzing data, for generating plots. And everything is free and open source. And so you can go in and you can say, okay, I like this package a lot. It's really great for what I'm doing, but I want to tweak it slightly. You can download all the source code if you want to. and make your little tweak and then recompile it, and voila, you have your own package that does your own function, and it works really, really great for your data. So that being said, you'll notice that the learning curve for those two programs that are kind of on their way out is definitely less steep than learning, for example, R, MATLAB, or SAS. So those are all language-based statistical softwares. So you're basically, if you decide to learn one of those programs, you're going to be learning coding. You're going to be learning a computer language from scratch. There are a lot of tools that will make it easier for you and sort of make it a lot more intuitive and a lot more user-friendly. But when you open up R for the first time, it's just a blank screen, and it's just waiting for you to type in commands similar to that in the style that you would see in like Java. So if you've ever played around with Java before, you can open up Java and it's just a blank window, and then you start typing code in Java. Same thing in R. All it gives you is just a blank screen, says, what do you want me to do? So it's definitely a little intimidating. If you don't have any program experience, or if you've never used that kind of statistical software before, R, Python, SAS, MATLAB, okay, they can be a little intimidating when you first start out. It's like learning a whole new language. It's like learning Spanish or French or German. You walk into it and you don't even know what the letters are, what the syntax is, how to phrase a question. You're completely lost. Fortunately, there's a lot of really, really good resources to help you get over those initial hurdles, and I've linked a lot of them in this presentation. So if you guys go on the presentation at the very end, there's a lot of links for places you can download these free programs and you can learn how to use them. All right, so I promised you guys I would talk a bit about power analysis. We're shifting away from which software to choose. So power analyses are definitely an underused tool, I think, in biology, but they're really, really useful. And like I said before, if you're working with any sort of lab animal in a controlled setting, you're going to have to do one of these for your IACUC approval. The power analyses are based on 4 variables. There's an intimate relationship between these 4 variables. The first one is sample size. That's what we're interested in determining. What kind of sample size do I need to run the experiment? Do I need to inject 5 mice with a drug, or do I need to inject 100 mice with a drug? The effect size is one it's a little tricky, but the effect size is essentially what kind of difference do you want to see in your response variable. So if I'm injecting mice with a drug and I want to see how large they grow, so say it's a new growth hormone for steroid bodybuilders. If I inject mice with a drug and they grow by 1%, that's not super exciting as far as a drug company goes, because 1% growth is not that much. So maybe I want to see a 10% growth in the mice. Maybe I want that to be my minimum effect size. So you can set that effect size in your power analysis. You can set the difference between your groups that you want to see. The significance level, if you guys have taken a stats class, you're probably familiar with. The significance level is denoted by the Greek letter alpha. In general, it's set to 0.05. I'm sure there's a really great reason why it is set to 0.05. I don't know why it is always 0.05. Sometimes it's less, sometimes it's more. But I would say in 90% of the biological papers you read, they choose an alpha level of 0.05. And then the 4th variable is the power. The power is kind of an interesting. It's a little difficult to conceptualize, but some of you guys might remember from your statistical class that your alpha level or your significance level is actually the same level as your type 1 error. Does anybody remember what a type 1 error is from stats class? Yeah, it's a tough one. So a type 1 error is the probability that you see an effect, but there actually isn't one. So say I set my alpha level at 0.05 or 0.05, and I'm doing my statistical test, and my statistical test says, hey, there's a your p-value is less than your significance level. Your test is significant. You hear that all the time in stats, right? The test was significant. Well, what that actually means is that there is a small chance that the phenomenon that you're seeing, say it's difference in mice size, one mouse got bigger than the other one, the phenomenon that you're seeing could have occurred by chance. It could have been totally random. It could have just been a random chance. And your alpha level is setting basically the thresholds of rejecting or accepting those results, but they were actually just random occurrence. So you say, yes, there was an effect of my treatment, but it was actually due to random chance and not treatment. That's a type 1 error. Your type 2 error is kind of the other error that you can make in statistics. So say I inject my mice and I get some of them that are slightly bigger than the others, but in my treatment group, but it's actually not statistically significant. If I say, okay, if there actually was no effect of the treatment, but there actually was an effect of the treatment, I just couldn't detect it statistically, that's what we refer to as a type 2 error. So all 4 of those variables have this intimate relationship, and if you play around with them a little bit, you can do something like this. So these are called power curves, and they're based on those 4 variables. So here we set the power level. So that's the 1 minus our probability of a type 2 error. And then this is our correlation coefficient. So this is basically telling us, that would be an example of an effect size. So we have 2 variables. We're trying to see how well correlated they are. You can see if they're really well correlated, 0.5 correlation coefficient, we don't need to collect that much data. If we're trying to prove that 2 things are correlated, and in real life they are really well correlated, we don't have to collect that much data to prove it. If they aren't really well correlated, so if they have a correlation coefficient of 0.1, we might need to collect 200 samples in order to prove that that correlation actually exists. And as you can see, if we start from a very low power level, we only have to collect a small number of samples in order, even at very low correlations. If we raise our power level very high, all of a sudden now we're collecting 1,000 samples, 5 times as many samples. So doing one of these curves, you can say, okay, I think my variables are related about this much or this much, and then what kind of power are you comfortable with? Are you comfortable with 0.8? A lot of people are. If you're comfortable with 0.8, you pick that curve and you go out to your correlation coefficient, and then it'll spit out a number. It says, okay, you have to collect 54 samples. And that's what you end up doing. That would be something you could say to IACUC. It says, my power analysis indicated that I needed 54 samples. I'm going to do 55 because it's a nicer number. And then IACUC says, great, that sounds good. But you do need to present all of those things to the IACUC committee when you do those types of experiments. So all of this helps you figure out how many samples you need before you sample. Even if you're not working with IACUC, it's a good idea to try out one of these power curves, just because you could save yourself a lot of time. If you're conducting an experiment, you're designing an experiment, and you decide you want to measure 1,000 samples, and you do a power analysis, and you see, oh, you know what? I could get away with 200. You just saved yourself, 80% of your time by doing one of these power analyses, and they don't take that long. They're pretty quick, pretty easy. So I would definitely encourage you guys to try out a power analysis before you do your experiment. So I'm going to touch on statistics a little bit, but not too much. One thing you guys might be interested in is doing parametric or non-parametric statistics. Parametric statistics make assumptions about the distribution of data in a population. If we took the height of everybody in the entire world and plotted it on this plot right here, it's likely that it would come out looking something like this. There would be a normal distribution. Normal distributions occur all the time in nature. Not as often as, it's not every time, but a lot of things are normally distributed. And that's just because there's a lot of people that are centered around the average. And we have a few really tall people, we have a few really short people, but most people are in here. So that would result in what we call normal distribution. Paranormal distribution is a picture of a ghost. So parametric tests are based on the assumption that the population that you're interested in, the data are normally distributed. So the heights of trees in the Fairbanks area are normally distributed. The weights of mice are normally distributed. If you do parametric statistics, you're making some assumptions about those distributions. There's a lot of pressure for you guys to use, well, I don't know if I should say pressure. There's a lot of movement towards using these parametric statistics because they're more traditional. We've been using parametric statistics for a long time. I would say they make up the majority of what we read in biology, especially in the life sciences. And they're maybe slightly more intuitive because they're based on comparing means. We're really, really comfortable with this idea of I'm going to take the average of this group compared to the average of this group, and if they're different, then I can say that this group is higher than this group. That makes a lot of sense. But there are many situations which require the use of non-parametric data, non-parametric statistics. If your data are non-normally distributed, if your data are sometimes if your data are categorical, if your data are ordinal, if your data are, for instance, if you're interested, has anybody ever heard of a chi-square test? Yep. So a chi-square test would be, let's say, for example, I have a bag of M&Ms, and I'm interested in seeing what the frequency of the color of the M&Ms are. So I can make an assumption, or I can make a hypothesis that, maybe there's the equal distribution of colors in a bag of M&Ms. So I pull out an M&M, I say, okay, it's blue, take it out. Another M&M, it's green. At the end of the day, I'm going to have a percent that were green, a percent that were blue, a percent that were red, so on and so forth. Actually, I don't remember the colors of the M&Ms. There's a brown one, right? I'm not just making that up? Yeah, there's a brown M&M. So at the end of the day, I have all of these proportions of these different colors of M&Ms. I would actually do something called a chi-square test, which would tell me whether or not those proportions are equal. And that is not based on anything, there's no assumption of normality. I'm not assuming that the M&M colors are normally distributed. That would be silly, because they're not a continuous variable. So if you guys have done, or if you're doing some sort of experiment where you are expecting some sort of categorical proportions and categorical variables, you might be utilizing these non-parametric statistics and you didn't even know it. Bayesian statistics, in my field, which is sort of marine mammal field, marine mammal toxicology in particular, there's been sort of this upsweep towards Bayesian statistics in the last couple of years. And you find people that are very pro-Bayesian statistics and very anti-Bayesian statistics. If you had asked me like a year ago what these statistics are, I would have had no idea. I definitely heard the term. I've read it in papers, and papers generally do a pretty good job of explaining what they're doing when they're using these types of tests. I'll give you a brief explanation right now. I'm not an expert in these types of statistics, so bear with me. But instead of being based on something like a normal distribution, Bayesian statistics actually make prior assumptions about the data. So if I make a prior assumption, so these priors can be based on what people have published in the literature, what you think is going on with the particular variable. Like I could say, if I'm interested in analyzing the tree height of trees around Fairbanks, I could say, you know what, I think they're all about 30 meters tall. There's going to be a distribution there, but more or less the average is going to be 30, and there's going to be this amount of spread. That would be my prior distribution. So you make these assumptions called priors based on kind of an educated guess, you might say. And suppose I go out and I measure all the trees, and they actually had a mean of like 60 meters. Turns out I was way off. That would be this yellow thing here. So this would be our what we actually measure. We don't just take this as writ that is the actual distribution of the population, because we made these priors, because we know that other people have measured this variable and they've come up with different numbers. But what we can do is we can adjust our distribution to kind of be a happy medium between those two things. So we make measurements, and then we adjust our priors into our posterior distributions. And that is everything I know about Bayesian statistics. It's not very much. But so my summary of Bayesian statistics is that you should probably know about it, especially if it's something that's common in your field. Are you going to use it if it's not common in your field? Probably not. I think I can go my entire career without getting into Bayesian statistics, but some people really, really like it. They're very passionate about it, and if you see it popping up more and more in your field, it could definitely be something you should look into. So here I have the slide on helpful tools for getting started. You can see I put R up here. So download the actual programming language. It's a very bare bones interface if you just download that. So a lot of people work in some sort of GUI. In this case, I really recommend RStudio. I think it's the one with the best support. It's actually got a lot of point and click features, which is really nice when you're getting started. And it's definitely got the most sort of user-friendly features. If you're interested in working with Python, is anybody doing like bioinformatics? Is anybody working with like gene sequences, DNA, RNA, something like that? Yeah. If you're interested in doing any sort of work with high throughput sequencing, so if you're doing any next gen sequencing like of DNA or RNA or something like that, it might be a really good idea, especially early in your guys' careers, to try to pick up something like Python. Python is used a lot for high throughput sequencing. So we use Python scripts for a lot of stuff related to gene sequencing. It's not, at this point, it's not particularly useful, I would say, in sort of just general biology. It can be used in much the same way that R is used, but again, it's not quite there yet. Maybe in another couple years there will be more tools available, but at this point, R is definitely the easiest language to learn and use in biology. If you wanted to continue using Excel, I'm sure most of you guys know at least a little bit about Excel. I included a couple links up here on creating good graphics in Excel, so how to choose a plot to represent your data, and then a little bit about how to do some statistics in Excel. So what we're going to do for the second half of the class, well, first I'm going to give you guys a break to get up and stretch your legs and go get some water. But in the second half of class, I put 4 data sets up on Blackboard. And these are data sets that are either publicly available data, or they're my data that I've already published on. So what these data are, I gave a brief description. The first one is actually air quality data from the Fairbanks North Star Borough. So if you guys have lived here for a winter, you know that in the wintertime or in the summertime, if there's a forest fire, Fairbanks actually has some pretty poor air quality. You'll notice that like in the winter, for instance, the ice fog kind of settles down in the lowlands. And what people worry about in those situations are what's called PM 2.5, or particulate matter of 2.5 microns or less. And this is actually air quality data from Fairbanks from July to August of this summer. And there's a couple other variables on there, including like wind speed, temperature data, ozone concentrations. And so those are organized by day, I think, or by hour, actually. By day or by hour. But yeah, so you have the PM 2.5 concentrations from Fairbanks from the summer. It's not a particularly smoky year, but I think there were a couple days where we had some smoke. The second data is some fisheries data from the Bering Sea. Again, this is publicly available data online. So there's fish data, including catch number, average size, and location for a couple of fisheries in the Gulf and the Bering Sea. So those data are represented as, they give you a species and how much mass each fish weighed was the total mass of all the fish of that species that they hauled in for that location. This is some morphometric and isotope data from fish from Prudhoe Bay. So if any of you guys have worked with stable isotopes before, I know Vanessa has. Stable isotopes are a great way to look at feeding ecology, try to figure out what an animal is feeding on and where it's feeding in either the nearshore ecosystem, the pelagic ecosystem, the benthic ecosystem. So there's some morphometric and data for fish that were captured up there. I can't recall which species of fish they were off the top of my head, but it's in the spreadsheet. And then finally, I have some data that I collected, which was actually total and methylmercury concentrations from sculpin, which are another fish species. And we measured them in different tissues. So we had 4 muscle tissues, and then heart and kidney and liver. And all of those are in the spreadsheet. We also have age and length, so how old the fish were, how long the fish were, what size they were. So those are all in there. And what I'm envisioning for this next hour is for you guys to sort of wade around in this data. And I realized that you didn't collect these data and that you didn't follow the first 8 steps before you actually, collected the data. But what I want you to do is sort of look through the data, maybe make a couple plots, if you can, in Excel or R, or whatever program you'd like, and just make an observation and come up with a way to plot it, come up with a way to make a graph that conveys your observation. So if you're looking at the fisheries data and you say, wow, they caught a lot more of this species of fish here than in this area, try to make a plot that communicates that. If you're looking at the PM 2.5 concentrations and you think, wow, I wonder if wind speed is at all associated with PM 2.5 concentration. I wonder if the wind affects it at all. Try to make a plot that investigates that question and then try to address that. If you guys are comfortable, if you're familiar with statistics, you want to try running stats on your hypothesis, on your observation, go for it. That would be awesome. But you guys have an hour to try to make a plot, and I'm going to be wandering around and help. Andres, if you can stay, we can wander around, and Teresa as well. We can help you guys, and I'm hoping that in about 45 minutes you guys can come up with a plot and be able to present it up here. So you can hold up your computer. You don't have to put it on the screen or anything, but just kind of hold up your computer and everybody can see. We can all gather around up front. Everybody can see kind of what you did, and you can give like a 1-minute presentation on it. Sound good? All right. So yeah, you guys can take a break for a little bit, and then if you need a computer, they're in the back over there. All the data should be available on Blackboard.